Great. Okay, let's go. So good afternoon, everyone. I'd like to extend a very warm thank you to all of you for taking the time to join us today, especially our panelists and speakers, but also to each of you watching in, tuning in online. We're great to see such interest. And as I said, please be sure to use the chat box to let us know who you are and where you're writing from. The Q&A box is there for your questions, and we will address them towards the end of the webinar. But if you have any questions in particular that would like to be addressed early, then that's fine as well. In particular, questions for Dr. Kayleigh Wyles, who will be presenting in a few minutes. She will not be able to join us for the roundtable discussion, so please get those questions in early and we will ask those directly after the presentation. So a special thank you to the BlueMissionAA team for allowing us to collaborate as part of their weekly hour series. This is the 5th episode on a second series of Atlantic Arctic Weekly Hours, so I would recommend you all go check out the other episodes. They are very exciting and very riveting. So this is the 4th of MAELSTROM webinars, and the point of these is really to share knowledge, to share and collaborate across projects, and really connect with society and with policymakers across the Science Policy Society nexus. So we're really hoping to engage with you all today and get some inspiring talks and presentations, and then have some further discussions later on. So the theme of today's webinar is One Ocean, One Health. So the idea behind this theme is really taking a collaborative, multidisciplinary and transboundary approach to our planetary health. Our global and interconnected health systems are very intricate and can be challenging, especially when you're taking in a highly variable ocean and waterway system. It is now known that the health of the environment is closely linked with the health of our society, and to manage pollution is to manage the risk of our exposure to harmful substances. The interconnected nature of human health, animal health, and environment health can definitely be challenging, but with collaboration, with empowerment, and with activation of our society, I believe we can really make a difference in this field. Our rivers and oceans are unfortunately where much of our waste eventually ends up. And now, especially during the UN Decade of Ocean Science for Sustainable Development, it's crucial that we remember the important role of our ocean in our health systems and the impact of our societal practices have on our environment. Today, I'm delighted to welcome a diverse array of scientists and audience members to explore this theme. I invite you all to think about how these disciplines work together. How do your projects or your work or your areas of interest engage with these? And what potential routes of collaboration or synergies can we build down the road? So we want to really spread awareness, and we want to have a positive effect on the policies and the practices that we all engage in on a daily basis. With that, I would like to invite my colleague, Dr. Luis Vieira, to take the floor and introduce the speakers. Luis is an ecologist and ecotoxicologist here at the CIIMAR Institute of the University of Porto. He spent many years working on the impacts of pollutants, especially marine litter, and microplastics on aquatic organisms. And I've had the great pleasure of working with him on the MAELSTROM project, playing a crucial role in the engagement of stakeholders, the public outreach and education engagements, and the implementation of the exciting bubble barrier, which you'll hear about later. So with that, over to you, Luis.
Thank you very much, Garry, especially for a very kind introduction. Also, hello and good afternoon to all the panelists and participants who are having an excellent participation and going up. And thank you all for your interest in this webinar today dedicated to the One Health perspective, which is also a mission from the European project MAELSTROM, contributing to tackling the increasing impacts of marine litter, in particular plastic pollution on both ecosystem and human health. And of course, a special thanks, acknowledgement to all the panelists, also leading academics, who today dedicated their time to present and discuss novel research findings and developments in marine and human health research. So we start now the presentation session with an overview of the MAELSTROM project to sustainability remove and manage marine litter by Dr. Vanessa Moschino. Vanessa is the co-PI of MAELSTROM project and a research scientist at the Italian National Research Council, working in marine ecotoxicology and biomonitoring. understanding the effects of multiple stressors in several aquatic organisms. Vanessa has an extensive experience in illustrating and characterizing microplastics for several environmental matrices, and she has and is contributing to several national and international initiatives and projects dedicated to this topic, and of course, including MAELSTROM. So welcome, Vanessa. Thank you for your time to dedicate your presentation to this webinar. So the floor is yours.
Okay, perfect. So in this presentation, I'm going to give an overview of the MAELSTROM projects that implements smart technologies for the removal and the management of marine litter. MAELSTROM in number, the project consists of 14 partners from 8 European countries. The project is an H2020 project and has a budget of 6 million euros and started at the beginning of the 2021 and will finish at the end of the 2024. So 4 years of project, and the partners are both belonging to private and public companies. And here, the MAELSTROM project tries to address all the different issues related to marine litter, in particular to identify the litter accumulation hotspots, then to design and implement innovative technology for the removal of marine litter, both from the seabed and the water column, and then to try to recycle all the collected litter and also to create new products and materials. And the MAELSTROM project has 5 main objectives. The first one is to assess the effectiveness and possible impact of the removal technologies on the coastal marine ecosystem. And in the framework of these activities, we selected 2 demo sites, one in Italy, and in particular in the Venice coastal area, and one in Portugal, in the Porto region, and it is the estuary of the Ave River. How we can identify marine litter sources and hotspot. We perform an extensive overview starting from the EMOD data related to the beach litter, and then we try to identify the different sources of this beach litter. We also implemented numerical models in both sides, and here you can see some figures of these models to identify the possible hotspots and accumulation site of marine and riverine litter. Once the demo sites were identified, environmental assessments were performed before and after the removal operation with the innovative technology. This is fundamental to understand and to assess the effectiveness of the technologies, but also to evaluate their impact and effect on the ecosystem. And here you can see some images of the multi-beam mapping performed in the Venice coastal area. before and after the cleaning operation. So it's during the cleaning operations, sometimes were removed, and here you can see the presence of tires in the bottom before the cleaning operation, and the absence of these tires after the cleaning operation. To assess the effect and presence of marine litter and microplastic in the two coastal environment, we apply this scheme of monitoring. In particular, we monitor macrolitter in the floating in the river, in the Ave River, and we identify the litter on the seafloor through visual census and also through acoustic remote sensing. We also perform an extensive monitoring of microplastic in different environmental matrices, such as water, sediments, and biota, to understand how the community can change after the removal technology. We also have done a lot of activity and sampling on the biological community to understand the possible alteration after the removal operation. During the activities of the project, we also define some important guidelines on how to consider marine litter pollution and remediator strategy in the framework of maritime spatial planning, and the report about this activity is public, so you can see it and download it. The second objective of the project is to implement the innovative technologies to remove the marine litter from the seabed and the water column. In the two demo sites, two technologies have been developed. The bubble barrier in Portugal, in the Ave River estuary, and the robotic seabed cleaning platform in Venice. And here you can see the pictures of the launch events in the two demo sites. The bubble barrier deployed in the estuary of the Ave River consists of a tube placed on the river bottom that create bubbles that catch plastic pollution in river before it reaches the oceans. It is also provided with solar panels and a catchment system to collect all the litter passing through the river. The seabed cleaning platform consists in a floating platform that supports a cable robot provided with cameras and a grip that can selectively pick up the litter from the sea bottom. And also the seabed cleaning platform is controlled remotely and has also a pipe that can suck up smaller litter from the seabed. Here you can see some photo of the cleaning operation performed in the Venice lagoon, and the seabed cleaning platform remove about 2,200 kilos of marine litter from the channel of Venice, and some examples are large metal structures and also boats, some tires, part of machine. But what we can do with the collected litter. The activity of the project are devoted also to feeding the circular economy and sustainability. Oops. Okay, so during the project, MAELSTROM important were developed to track the marine litter collected during the cleaning activities and to follow their fate, starting from the collection and finishing from to the recycling of these items. And all the steps can be monitored with the MAELSTROM app and through the portal. We also, the partners of the project, also design and implement an AI-driven robotic system to identify, segregate, and sort the different collected items, also considering their the polymers that are made of the plastic. So it's very important, this part, because one of the main issues is to try to separate correctly the collected item and identify the polymers of the items. One other part of the project consists in transform the marine litter in new products through mechanical and chemical recycling. Here you can see an example of the mechanical recycling. The litter collected were then sorted and shredded, and new panels were created. These panels are used to make new furniture, such as the bench and containers, and all the process is certified. So it's possible to certify that all these furnitures derived from marine litter. Through the chemical recycling, and in particular through the low temperature pyrolysis, is possible to obtain from marine litter marine gas oil, marine fuel, and also other chemical that have values, economical values. Here you can see the different step of the process. So the use of non-selected and non-sorted marine litter to produce the oil. One other activity of the project is clustering of blue technologies for joint plastic strategy oriented effort. And only a few words about this, because I think that there is another presentation. And the project did and is doing a lot of activities to reach this aim, and in particular one of these is the creation of a MAELSTROM thematic working group on marine litter and on the EU plastic strategies, and several webinars were organized and to tackle this issue of the marine litter. Also, the MAELSTROM project contributed to the EU mission Restore Our Oceans and Waters by 2030. MAELSTROM was already part of the Blue Mission Med initiative. MAELSTROM participated to the first meeting of the EU mission and also to the high-level event organized in Palermo in May 2023. And the project also endorsed the mission charter. A lot of activity were performed to raise awareness of citizens and also of younger generation to sensibilize them in the prevention, removal, and circular economy of marine litter. MAELSTROM organized a lot of cleanups, not only in the two demo sites, but also in other places. Also participate to the Anthropocene exhibition held in Venice in June and from June to September 2023. And MAELSTROM has a collaboration with the Venice Art Biennale. So, summarizing all the activities performed in the framework of this project, the project developed 6 technologies at the tier 7. The seabed cleaning platform removed about 2,200 kilos of marine litter from the seabed, but also during the cleanups performed in the beaches, the MAELSTROM teams and citizens collected about 7,300 kilos of beach litter. 2 removal technologies, innovative removal technologies, have been developed in the framework of this project. One of these is already patented, and also we supported 10 MSc theses and 2 PhD, and a lot of the data produced in the framework of the project are fair data, and we also did 7 research objects on these data. So thank you for your attention.
Thank you very much, Vanessa, for a very interesting presentation showing how MAELSTROM is already implementing innovative solutions which in fact are contributing for cleaner future and of course better human health and ecosystem health. As you know, in addition to technological solutions, we also require societal solutions to encourage a future project, positive action. This project, MAELSTROM, emphasizes the participation of both local, regional, national, and international stakeholders, facilitating the long-term adoption of the technologies that were presented by Vanessa. And this collaboration will also contribute to upstream initiatives aimed to address the marine litter issue. In this topic, Professor Isabel Sousa Pinto will present the importance of stakeholder engagement in marine litter management. Isabel is a professor at the University of Porto and leads the Aquatic Biodiversity Group at CIIMAR, also being a member of the board of directors. Her research focuses on the marine biodiversity and on ecosystem functioning, with a particular interest and focus in science, policy, society interface, also contributing to ocean literacy. She is serving in different European and international steering committees, for example, Euromarine and Lighthouse, and Isabel is also the co-chair of Marine Biodiversity Observation Network. She is also an elected member at the Intergovernmental Panel on Biodiversity and Ecosystem Services Multidisciplinary Expert Panel, also being a co-chair of its Knowledge and Data Task Force. Thank you, Isabel, for your contribution and time. So we are very interested to see the presentation.
Well, thank you, and thank you for inviting me to this webinar. And I'll be very short because Fantino already gave a very good overview of this project. But what I'm going to do is really focus then on what we did and what we learned working with different stakeholders. So, of course, it's, as Luis said, absolutely essential. We can do a lot of research and also have wonderful technologies, but we need also to engage and to really bring all sectors of society to this fight against marine litter. And so when we're trying to do this, apply these solutions, we really need very much to include a set of stakeholders. Some of them we learn are completely vital. Others are, it's good to involve them. This will increase the impact of our work. So, of course, the local administration, especially if you have a technology that is going to stay in a place, so it's going to be part, as a fixture, is going to be part of the city, of the community. Of course, we need to involve them from the start. We did that to really engage them and also to hear what they want to, what they have to say and how they want things done, to understand their interests and problems. So it is really vital, but also we involved also regional, national, and even international. Since we are an international project, we also involved different administrations. Of course, we had to involve, and I'm presenting this because I think this is not just for this project. I mean, these are more general issues. So we had to involve a lot of administration also at local level. So the captaincies, the harbor masters, environmental administration, both at local and national level, companies that do the waste management, recycling, engineers, local commerce. And then, of course, the specific users, the people actually from the general public, but the people that use this environment, the fishers groups, the water sports groups, recreational users, NGOs, and so on. And of course, we also involved, and we think it's very important to involve also people engaged with education and really work with the schools, but also with the civil society, to strengthen the ocean literacy. I mean, all the knowledge about what needs to be done and also what are the impacts of this litter in our environment. And then to develop with them science citizens initiatives. That's what we're doing too, and really build public interest. So the goal of this in the end is to co-develop these solutions. So we have, of course, we have technologies that we want to promote, but in the end, we need to co-develop them with especially with the local communities, to have buy-in and what many like to call now a social license to operate. So to have their acceptance and their goodwill to our initiatives. So what we did in MAELSTROM was to engage at all levels, as I said, and many sectors, from local to international. And the idea was to share experiences that we all have regarding this problem of marine litter, but also to disseminate ideas that were created by the project and practices. And we did this in different international meetings. I'll be talking very quickly, but then also engaging to policymakers to understand the needs and interests. And this is also from local to international level. And then with all this, also to co-design and complement the solutions. Of course, this is basically, and is more direct with local stakeholders, but also the co-design can also be done at different levels with different stakeholders. So we have a very, very wide range of collaborations from other projects that I think is very important to collaborate with other projects that are ongoing and just finished, and they produce results that are relevant for our work, but of course also with all the municipalities and the different sectors from society to global networks like the MBON, but also with these organizations, the United Nations Decade, and so on. So just very quickly going through some examples. What we did is really, we really meet a lot with the local stakeholders, so both the administration, but also these groups that I mentioned. And we had more than 30 meetings during these years with the municipality and so on to make sure that we co-develop these solutions with them, and then when they are in place, that they will actually then own them. So the idea for the bubble barrier is that the municipality of Vieira will own it at the end of the project. So this is not easy. There was a lot of issues and barriers and that we had to discuss and that we have to agree on. And so this was really very, I think is an example of the things we need to do if you want really to implement these solutions, especially those that are more permanent. Then we had a lot of work done also with the different sectors of the community. We had this inauguration, of course, with our Secretary of State of the Sea, so the person in charge at the national level of the marine affairs. But we have, of course, the impact assessment that is very interesting for the municipality to know what the impact will be from this. We are still working on this. What is the impact, the real impact that this technology will have, so the bubble barrier will have in the river ecosystems and also on the coast. We are also characterizing the litter, which is very interesting also for the authorities, so they know where this litter is coming from, and then they can act on this to reduce actually the arrival of this litter into the rivers and the ocean. And then, of course, since we have been working with the local authorities, they are expected to take over this after the end of the year. So we also did a lot of work with the citizens. Vanessa already mentioned, I just mentioned three different examples. So the first one that was done in Matosinhos, at the time we did not have a decision where this technology was going to be placed. We did not only a beach cleanup, but we also did a presentation of the technology, and we discussed and we had some presentations about the importance of reducing marine litter. Then we had an innovative event in Vila do Conde where we put together this beach cleanup at the end of the afternoon with the observation of the stars. This was so interesting that the local municipality is going to do it every year. So this is going, and they will took over this initiative. And we had, of course, the Venice cleanup that Vanessa already mentioned, where there was a lot of garbage taken out with a lot of different groups from society that are interested in this kind of work. There's some numbers, just we did some survey of the, so our members of our society gave some feedback. We got 40 from the more than 100 that participated in this Sunset Beach cleanup to give us some feedback of what they thought. And what was interesting, they were very happy and proud of participating in this work. The 100% will do it again. They show those that respond to our questionnaire were quite knowledge about the impacts of marine microplastics and so on. Of course, they didn't know the technology. The technology is new. But what I thought was interesting is that, well, they thought that there was a lot of litter in the beach and especially in the river. But what was interesting when you ask who should be the main responsible to deal with this problem, they thought the EU government. I thought this was curious because that will not be my first guess. But then we also did a lot of support for disseminating these technologies by live demonstrations of the prototypes for many different events, both national, international. We had a very strong participation in the UNOC event here in Lisbon for the UN Ocean Conference. We had several events and we also had the presentation. And so also during the beach cleanups, we had in some of them this presented so people could know more about this. And for many of these reasons, we were awarded actually this Clean Coast and Healthy Seas Atlantic Project Award just a few months ago. So we know already Fantina said that we endorsed the Mission Ocean, but we've been present so in many of these organizations from Mission Blue Med, but also from the European Ocean Days, again, demonstrating the technologies and presenting also. So we also organize workshops. This is a more technical event where we brought together different people with different knowledge, not only technology, but also knowledge from society to tackle this problem of litter. So we had two in Venice, one the Ecomondo, and we'll have more until the end of the project. And then we had these webinars. This is the fourth. We had several others, different themes. And what I would like to end up with is this forum that Vanessa also alluded to, this working group. So we are bringing together all this information that we got from all these activities with stakeholders, and we are creating this working group too. So the main objective is this publication and recommendation knowledge gaps in marine litter management, but also to collect what we told best practices. I think we should talk about good practices on all these sectors. So good practice on collecting the marine litter, on recycling, on engaging society, on preventing the litter to get to the ocean and so on. And we, of course, I invited those interests to participate. And I end now. Thank you very much for your attention.
Thank you, Isabel, for your very interesting and engaging presentation, showing in fact, the key role of multi-sectorial stakeholders engagement, in fact, very linked to the next presentation. No matter how successful these technologies and the solutions are, we need to connect, in fact, society with nature, communities with ecosystems, and co-design effective and valuable solutions. That's the key for the success of these implementations. Dr. Kayleigh Wyles will highlight the human dimension of marine litter, from societal impacts to the solutions. Kayleigh Wyles is Associate Professor of Environmental Psychology at the University of Plymouth. She's a well-renowned international psychologist whose focus in terms of research is mainly dedicated how people interact with natural world. Kayleigh is also collaborating with a variety of stakeholders from NGOs to advising decision makers, such as contributing as advisor at the United Nations Joint Group for Experts on Scientific Aspects of Marine Environmental Protection. And of course, with a more recent focus on the Scientist Coalition for an Effective Plastics Treaty. So welcome, Kayleigh. Thank you for accepting this invitation. So the presentation's floor is yours.
Brilliant, thank you very much. And yeah, thank you for the invitations. I'm sorry that I can't make this later hour due to changes of times. But yes, today I'll be focusing a bit more about that stakeholder human dimension side of things. And start with a conclusion, basically. As we already know, marine litter is purely a human issue. It's unlike other socio-environmental issues, it is caused exactly and only by people, but we are also impacted by it, and we also have the power and responsibility to address it. So in these 10 minutes, I'll try to summarize each of these, reflecting on a number of different projects I've worked on. So first of all, we contribute to the problem. We already know that plastic pollution and marine litter can happen from different aspects of the supply chain. And behind each of these stages are people, people's behaviours and people's decisions. And this can be different sectors, but also different decisions throughout it. So, for example, if you just think about one single product, a plastic bottle, how you decide to make it will influence how sustainable it is. So do you use brand new raw virgin materials to make it nice, clear and transparent, or do you use recycled materials? Next question is then, how do you design it in terms of, do you want it to be a colourful bottle so it stands out on the shelf, but then it can't be recycled in the same ways? Then of course, when it comes to use, do you buy it every day, buy a brand new water bottle, or do you reuse it or repurpose it in some way? And then finally, disposal. What do you do with it? Do you litter it? Do you recycle it? Do you put it in normal waste? All of these decisions influence how sustainable the ultimate product is, and therefore whether or not any parts of it leaks into the ocean. So behind all of this are people. And what we need to know is what people do, who are the key people and actors, stakeholders, but also why, what drives those decisions and behaviours. So just focusing on one example when it comes to waste management, we did a project in Norway and Svalbard looking at people's self-reported behaviours when it comes to waste management. And we looked at lots of different things to try to work out what are the strongest predictors of these behaviours. And yes, we found that there's some differences in terms of demographics. Females tend to do a bit more, people with children a bit more proactive, and so on. But what we find is it's more the internal, intrinsic reasons which influence us. It's our attitudes. So what are our views on plastic pollution? What do we think other people are doing? That has a big influence on what impacts our behaviour. And do we even feel like we have control over it? And these are much stronger predictors than, say, knowledge. Just knowing about the issue isn't enough. You need to care. You need to have an attitude or have strong feelings about it in one way or another. So this is just one example of how we contribute to the problem and understanding not only who is doing it, what are the key behaviours, but why. And then ultimately, once we know why, we can hopefully address that with some behavioural change solutions. Going down the question of why, we can also talk about that we as humans are impacted. And I know later on we'll have a session about other physical health impacts, but there's also mental health impacts of seeing and litter and marine litter. So one study that I did a few years back was I showed people lots of different pictures of beaches, some which were clean or had natural debris, some which had types of litter, either fishing related litter or public related litter. And using quantitative survey questions, I managed to show that the clean and the natural debris was seen as positive. People liked it, they felt happy if they were in this environment, they thought it'd be a relaxing environment to be in. But as soon as litter was present, that took away all those normal benefits. And what we found with our qualitative open-ended questions is that there was a reason behind the difference between public-related litter and fishing-related litter. So you're seeing fishing-related litter, yes, it was bad, it wasn't as nice, but there was some form of, well, it's a secondary byproduct of marine industry. Whereas as soon as you see public-related litter, it had a lot more negative connotations. It showed that people were being disrespectful or careless or selfish, and that had a knock-on impact on our mental health. But as well as having direct mental health impacts, we can also infer that there's going to be indirect impacts on society. And so this is through the use of ecosystem services. So with this paper, what we did is reviewed all the literature which was out there at that moment in time in 2016, which was over 1,190 studies. And we looked at what are the impacts on different subjects being people, but also different animals and different ecosystems. And we could see that for most, this was having a negative impact. Plastic pollution was harming. We know already from things like ingestion, entanglement, and rafting and stuff like that has a negative impact on these subjects. But we then wanted to see, well, what does that mean in terms of ecosystem services and the benefits that we get? And when looking at all of the different evidence out there, just in summary, we found that on the whole, there's lots of negative impacts. So just thinking, if it's having an impact on the fish that we eat, that's then going to affect our food chain. And also the cultural benefits, a bit like what I've just mentioned beforehand. And within this paper, we then converted that into a social cost of plastic to, again, hopefully engage different stakeholders to show a comparable financial cost. And so we know that marine ecosystem services are valued around $50 trillion per year. But if we're seeing a reduction in those services because of plastic pollution, based on the estimates on how much is being released into the ocean, we worked out that this would cost about $3,000 to $33,000 per ton of plastic each year. So we are impacted by plastic pollution, both directly, but also indirectly through these ecosystem services. So to move a bit more on the positive side of things, rather than doom and gloom, we also have the power and arguably responsibility to address it. Yes, this needs to be done through reducing the amount of plastic we're producing and releasing into the environment, but we also need to do cleanups, like was mentioned in the past two talks nicely. So beach cleans. With this, I then looked at the impact this has on not only the environment, but also on the people. So I love those findings that you just presented. And so with this, I've done numerous studies, but this one example, I looked at beach cleans and looked at change in people's mood, in their knowledge, but also their pro-environmental behaviour. And to see if it is unique to doing a beach clean compared to, say, just going to the beach for a coastal walk, or to do another citizen science element where you're recording biodiversity, rockpool rambles, I compared 3 different activities. And what we found here is, yes, this helps to clean the local environment. It also produces incredibly valuable data that we can use in science and policy making, but it also has impacts on people. So what we found is that it didn't really matter what you did. All 3 of these activities were found to be pleasurable. People enjoyed it. And they also reported being more pro-environmental as a result of it. So just by being in the environment, you're thinking, yes, I need to protect it, and therefore I'll change my actions. When it came to the citizen science element, we found that people's knowledge or self-reported knowledge increased. This wasn't similarly shown in the objective measure. We did a test and they didn't perform any better, but they were feeling more confident in their knowledge. And then what's unique to beach cleans was that they found this to be a much more meaningful and purposeful activity. And this is a key finding when it comes to the well-being benefits. But they were also likely to engage in another beach clean in the future. So by just doing this little taster, they are more likely to sign up to another beach clean in the future. So just to conclude, humans are 100% responsible for plastic pollution. This does have direct impacts on us through mental health, and we hope hear later on about other impacts, but also indirectly through ecosystem services. But on a more positive note, by actively engaging in this issue, not only does it improve the environment and that side of things, but it can also have direct benefits to those involved as well. So it's hopefully seen as a win-win. So going forward, from the human dimension point of view, I'd say the key things that we need to think about in our next steps is to understand who are the most impactful stakeholders and what are the key behaviours so that we have the greatest impact, but also what are the key reasons, why are people doing it that way, and therefore we can then design those interventions or messages accordingly. We need to understand the impacts to everybody, including people, to make it relevant to them. But when it comes to designing interventions, we also need to look at the impacts on the environment and on us, and the direct and indirect. It might be that you plan for it to have an impact on one thing, but it might have knock-on impacts, and we need to assess all of that before we can really fully implement interventions. So that's hopefully a bit of a whirlwind tour of some of the human dimension aspects when it comes to marine litter. I didn't do any of this alone, so thank you to my collaborators, and thank you very much for listening.
Wow, thank you very much, Kayleigh. That was really interesting. I think I can speak for everyone and say that was a very thrilling presentation. I really love those results about the psychology of the beach cleans. And I remember reading that 2016 paper and saying, wow, that's so interesting, how the beach clean can actually affect somebody's mood, and especially if it's a pleasurable, enjoyable activity, and it can have a really positive impact and knock-on impacts. So I wonder if it's time for a quick question for you now to off, and then we'll leave you go. I suppose in terms of that, and in terms of capitalizing on this knowledge that positive environmental behaviors can have more positive environmental knock-ons, where do you think we need to focus our attentions in the research dimension on that? What are the major knowledge gaps or themes that we should be exploring in the coming years down the line to really hammer home this information and utilize it to its best? So let's see what your take on that.
Yeah, I think that's a good question. And I'd say from a multidisciplinary point of view, we need to be thinking and looking at the impacts of not only the issue, but also those interventions. So something that I'm starting to explore, and I think there's a lot more that needs to be done, is any type of intervention, including technological ones. What are the knock-on impacts on people? So for example, if someone sees that there's a technological device cleaning up the local estuary, are they going to then think, oh, it's solved, I can add my rubbish there, it's a rubbish bin, I can just continue to litter because it's being dealt with elsewhere. So we need to look at the impacts on people, people's behavior, so that we don't actually encourage the opposite of what we're wanting to do. And also then look on, like when we talk about the impacts on the environment, does that then have knock-on impacts again on our health and well-being benefits? Are we, is it a win-win for everybody, or is there going to be some cost to society, or to financial costs, or to the environment? So I think that's probably it, is that taking that holistic approach, using different disciplines to really fully understand. So I love the work that this project's doing, fully understand what are the impacts to all these different groups.
That's excellent. Thank you very much. I think that's an area we've definitely talked about ourselves, but down the line, I think we'd love to bring in a little bit more of this environmental psychology into the field to understand these real impacts. So thank you very much.
Yeah, thank you very much, and sorry that I have to dash.
It's been a pleasure. So looking forward to seeing what our next speakers have in store. So Luis, over to you.
Yes, so we're moving now to the macro world, to the micro nanoplastics. As you all know, these tiny particles are present in the environment in the several matrices, posing significant risks for ecosystems and also for humans. In fact, recent research indicates very interesting results, but also concerning results about ingestion and their presence in food, water, air, so raising concerns about the human health also. So these particles are known to accumulate in organs, they can increase oxidative stress, and they absorb harmful chemicals, disrupting hormonal balance in case of the humans, and leading to long-term health complications. In this topic, the research work of Dr. Raymond Pieters is focused on understanding the immunotoxicological effects of these substances in food, airborne particles, and drugs. Raymond Pieters is Associate Professor at the Department Population of Health Science at the Institute of Risk Assessment Science IRIS, so in Utrecht University, Netherlands. And he is currently the principal coordinator of a very interesting project, POLYRISK, which investigates the impacts of microplastics on human health. So, Raymond, thank you for accepting the invitation. Yeah. So, thank you, and the floor is yours.
Thank you. Can you see my slides?
And not in the presentation mode.
Okay, try again. Now it is, right?
Yes, perfect.
Thank you. So, thank you to the organization for giving me the opportunity to talk about the POLYRISK project. And the POLYRISK project is already ongoing for 3 years now, and the aim of the project is to develop tools and methods and information to work or to design or develop a risk assessment framework for human health effects of micro and nanoplastics. And here on the first slide, you see that I'm not on my own. There's a lot of institutes helping us, helping me. We help each other actually, because it's a very complicated subject, as you can understand. I also like to acknowledge the Dutch project Momentum, which I'm also part of. So, and these projects, of course, work together to solve this issue. To start off with, this is a picture I took from the paper below. And it is just to illustrate that we are exposed to plastics, possibly exposed to microplastics via different routes. Inhalation is an important one, ingestion maybe also, and dermal can also be an entrance route. What is mentioned here is very important, that microplastics are really complex substances. There's a lot of additives in microplastics. It's up to 40,000 different chemicals that can be used in microplastics. So it's not only the particles that we are concerned with, but all the chemicals that are in the plastics as well. And these may have all kinds of effects. The best known maybe is endocrine disruption. Think about the phthalates and bisphenols that are present in microplastic or in plastics and may leach from the plastics. Another issue is that these plastics, if they are in the environment, they change, they age, and they may be modified on the surface. But they also may attract microbes, or they may combine with microbes and help microbes maybe to enter the system, or they may influence the microbiome in our gut. We, as POLYRISK, are mostly interested in inflammation as a form or a sort of immunotox effect. And inflammation, as you can imagine, is involved in many different diseases, allergies, autoimmune diseases, think about colitis, COPD, etc., etc. So inflammation can impact many adverse outcomes. This is the picture we work with, and we are working on methods, as mentioned, to assess the hazard, the exposure, and with that, the risk of these micro and nanoplastics. And this is just one slide on the immune system. It's maybe a little bit complicated still for those who don't know anything about the immune system. But the main message here is that we have an innate immune system and an adaptive immune system. And we focus on the innate immune system because that's key for inflammation. Every immune response starts with an inflammatory or a response of the innate immune system. And we focus, for instance, on epithelial cells, which can release all kinds of products that stimulate immune cells and also defend our cells against all kinds of microbes. And another important cell type is this dendritic cell, which translates signals from the innate immune system to the adaptive immune system. And also macrophages are key in this. Macrophages can take up all kinds of particles, including plastics. So they may have a role in this. This is all you need to remember. Macrophages, dendritic cells, and because I will show some data on these cells. So this is the first data slide. It's a lot of information maybe, but what we show here is the effects on macrophages of a different set of micro and nanoplastics. Here they are mentioned: polystyrene, PVC, polypropylene, polyamide, and titanium oxide as a nanoparticle, so the reference particle. We also, in this case, always use a microbial component, LPS, because it's a positive component. And here you see in the cell, we measured mitochondrial activity, we measured membrane damage, and this is measured by an enzyme that appears in the cultures. So this is in vitro effects. And we also looked at pro-inflammatory signals. One is a translation factor in the cell, which activates genes, among others, genes that have a pro-inflammatory effect. What you see here, these are virgin particles, not modified by chemicals or microbes, or just virgin particles. And what you actually see, that these particles have only at a high dose, and this is really, really high dose for our culture systems, of 100 microgram per ml, then we see an effect on mitochondrial activity, which is reduced in the darkest blue, darkest red bars, and the leakage of cells is increased. So that matches clearly with the two of them, but we don't see any inflammatory signal. So for that matter, we think, well, these virgin particles are not the most hazardous part of the sort of particles. So we thought, well, this is maybe not the real life particles that we are looking at. So then we were able to take other particles, which is polypropylene and polyethylene, but these were sort of modified chemically. And what happened is that these particles had a reactive group on their surface. And the reactive group is a double O bond, which can also be generated when you swallow the particles. If you have the particles in your gut, they may also be modified, and these reactive groups may appear on the surface of these particles. And this is the same marker we tested before, the NFKB, which was on the previous slide, where we didn't find anything with the virgin particles, and also not with polystyrene, which is the same particle we used before. There's no effect on NFKB. But if we use these modified particles, we see a clear response, indicating that chemical surface modification may render particles more immunoactive. We, as a check, also looked whether this bacterial component could be important for this. It's LPS. And for that, we compared LPS with the particles, and we also had an antibiotic here. This antibiotic is polymyxin B that can catch LPS. And if LPS is combined with polymyxin B, you see that the response is completely reduced, which is not the case for these two particle substances. So indicating that indeed it's not, in this case, not the microbial component, it's really the particle itself. So this was important information to us, and it even can be extended with the next set of data, where we had particles that we left in the River Rhine for a month in sunlight, well, only daytime, of course. And then we collected these particles again, and these weathered, so-called weathered particles are here in red. And we can just look at one of these graphs. You see that only the red particles that have a certain small size of 200 nanometers give a stimulation of this receptor. This is a receptor that appears on these dendritic cells, these key cells that translate information from innate to adaptive immune system. So we saw an upregulation of these receptors. And if then we combine these dendritic cells with T cells, which is adaptive cells, then we saw an increase of this proliferation of these T cells, which could indicate that dendritic cells activated by particles can activate T cells, and the T cells may activate allergic responses. So for us, this is an alert, a hazardous alert that something happens in these dendritic cells that may contribute to disease later on. Here you see the different particles in the cells, and it's important that I show this because then we know that indeed these particles are within the cells. They are taken up. So these data to us point that the virgin particles, freshly made, are not that dangerous to us, but as soon as they have a change in the surface, chemically or whatever, how it happens, or whether they contain environmental components, such as bacterial components, then they may be dangerous to our health. Now, what I cannot tell you much about is the studies we also perform, which is actually the cherry on the cake. We are now performing human studies in which we test both exposure to particles and the effect on the immune system in the volunteers or the workers that are exposed to the particles. Currently, we have finalized the textile fiber workplace exposure scenario, and we have finalized the urban outdoor air exposure scenario and the indoor soccer scenario. These studies take so much time. They all have taken 1 year to fully complete. We are now working on the data, and we already saw that in the ambient, the outdoor air, in case of a busy traffic situation, a stop and go situation, that we have much more rubber in the air. We of course know that air pollution will affect our lungs, but now we are interested to what extent is that due to rubber particles. The same is of course true, the same goal we have for the textile industry scenario and the indoor soccer scenario. So maybe in a half-year time, we can show you what comes out these studies. We are also working on the drinking water scenario, and there we face an interesting issue, and that's here. So what we now find in our own study is that we have 15,000 microparticles per liter. That's what we currently can identify in normal drinking bottles. In this new study here below, which came out a month ago, they looked at the nano size fraction, which is a very difficult thing to do. And they have a special machine. It's a very well done study. If you are interested in chemical analysis of particles, this is really the top-notch work they do. They look really at the nanoparticle size and found 240,000 nanoparticles per liter. This really sounds a lot, but if we compare it with what we do in our in vitro studies with polystyrene particles, for instance, that we use in a dose from 10 to 100 microgram per ml, this equals to 2.3 times to 10 to the 10, which is much more, by far much more than we find in drinking bottles. In a way, this is positive. I mean, that would mean that we can still drink water, but we don't know the long-term effects. We only know the in vitro effects. So I don't know, we cannot say now what happens if you drink your drink from drinking bottles all your life. So that is still a challenge, but at the moment, based on these studies, we cannot ethically not start this study because I think we are far too low in the exposure with these virgin particles in the drinking water. But it's good to know that I think that normal drinking water, if you take a fresh bottle, it may not be that hazardous to your health. Okay, summing up, we have seen, and it's not only our studies, that in the case of micro and nanoplastics, it's not only the particles, it's also the chemicals that are in the particles, and it's the chemical modification of the surface that makes a difference, and of course, the microbial contaminants that are on the particles. With that, we have still a lot of challenges, and the biggest challenge for us is how to detect and test micro and nanoplastics, because they are so small and so complex in composition. So we cannot really identify the lowest particles in our blood. It's too small. Only this one study that I showed you before is possibly able to do it. How we do it now is we just take from the blood, we sieve whatever we can from plastics, and we use we use a pyrolysis GCMS and identify the polymers, but we cannot see the particles. That's one thing. We are still working on what characteristics are relevant for exposure and hazard, and I already mentioned the surface chemistry, but also the shape and size may matter, and of course the chemistry that of the leachates from the micro nanoplastics may determine exposure and hazard. I briefly mentioned that we cannot say much about prolonged exposure. We know that acute exposure may have an effect if the particles are aged. And of course, what I mentioned, I think we really have to focus on this age and weather particles. So imagine in the case of this maelstrom, it's about the particles you find on the beach or in the seawater. To sum up, micro and nanoplastics are complex, as mentioned. Virgin particles seem not to be very effective on the immune system, but rather weathered and aged particles are immunoactive. But regardless of whether we find an effect on these virgin particles, I would say, well, we should also consider the precautionary principle, because it's not, as we all acknowledge, I think, that we should not spill the microplastics in our environment. And this is the group I work with, and I'm also part of CUSP, and these other 5 projects, other 4 projects, we work closely together with. So thank you for your attention, and I hope to get some questions later.
Thank you very much, Raymond, for a very, very interesting presentation, and also with some scary results about the presence of these particles in the water, in the rover, in the air, that we faced all the day, all days in the traffic, and the expected impacts of these particles in human health, in particular, some evidences of long-term effects in health. So recent reports also underscore the importance of strengthening synergies between multiple actors and effective outcomes. And these publications highlight several key topics, including identification of best practices, existing gaps, methodology standardization that we still need, and effective strategies for marine litter removal, recycling, and marketization. So next, Dr. Rudolf, thank you. Thank you, Raymond. Dr. Rudolf, in this topic, Dr. Rudolf Reuther will present the important lessons from microplastics research for standardization, policy, and the regulation that meets, in fact, the urgent gaps that are identified in official documents. Dr. Rudolf Reuther is a senior researcher and consultant. He is the head and founder of Environmental Assessment, the ENAS, and he has been working as a researcher and consultant for more than 30 years in the field of environmental monitoring, hazard identification, exposure analysis, and the risk assessment of several chemicals, including the adsorbing particles to microplastics. Rudolf has realized and managed many national and international research and contract works with several clients from mining industry to chemical industry, with a special main focus on the environmental assessment of facility, products, and plants. He is currently the scientific coordinator of another very interesting project that is the PlasticsFatE, that is designed to improve our present understanding of the impact of both micro and nanoplastics, including the associated additives, so that adsorbed contaminants that can be very harmful for human health. So thank you again for your time. So the presentation floor is yours.
Thank you, Luis. As I could have maybe the first slide already. Yeah, just to give you an overview on PlasticsFatE. This is the project I'm talking about in this presentation. And we are a rather big consortium of 28 partners from 11 European countries. And our duration is rather similar than MAELSTROM. And we are part of this CASP cluster, research cluster, European research cluster, which also POLYRISK and the other projects are included. Next slide. Here I just give a short overview of our goal, and our goal is to improve the current understanding of fate and effects of micro nanoplastics and those chemicals that are associated with these particles in the human body. And we have an approach that is based especially on a methodological aspect, namely to establish reliable methods for the detection, identification, quantification of these micro nanoplastics in complex human health and relevant matrices. And based on this, to be able to assess exposure, fate, and effects of these particles in the human body by means of in vitro, in vivo, in silico, and field studies. And we expect results that include new data and new insights on the impact of micro nanoplastics on human health, and also to develop new integrated human health and environmental risk assessment strategy. So that is really integrating both the environment and the human health, and based on data that is validated and that is generated by validated methods. And finally, of course, and that is the ultimate goal of all the research we are doing, be it as basic as and fundamental and new insights producing as it is, to really support policy, research, regulation, and standardization. Next slide. Just an overview of how the project is structured. It is structured into 5 vertical work packages, starting with material selection and physicochemical characterization methods, and from there going over to fate and exposure assessment and hazard assessment. And this work package 4 integrates everything in some risk assessment strategy. And of course, data management is something that is very important, because we have to make the data fair and open for stakeholders that are our final end users. Next slide. So you may know this figure, which has been published by this WHO report, which makes again more concrete or understanding how much hazard and exposure depend on the particle properties, and especially when they are at the micro and nanoscale size. So just to keep in mind, and of course, this may not be an exhaustive list of properties. Meanwhile, I think we in CASP, we could add some of these characteristics, even some more. Next slide. But this was also in a way our starting point, namely really to knowing that the different properties of particles are important for the effects or the exposure and the fate we will later on measure. we have to try to find relevant particles, relevant that are environment, that you can find in the environment, but also that are relevant from an economic point of view, to really get a good panel or list of test materials that have various properties, and so really get our methods validated and robust for characterization, but also for assessing exposure and hazard. In PlasticsFatE, as you can see, we have developed more than 20 various different types of test materials that have that consist of different polymers, and also that are derived from plastic items such as packages or bottles, plastic bottles, that have additives or not, and contaminants, and that have, of course, different sizes, shapes, and are also not pristine anymore, but has been aged and weathered, as we can find them out when we go out into the environment. We have also done interlaboratory comparison studies. These are comprehensive studies among the different laboratories that all measure a particular property of a material, and later on compare their results to see how valid or how robust the method is we are using in this context. And we have also done this already within VAMAS, and VAMAS has a specific standard for has the aim to develop a standard for the analysis of micro nanoplastics in the environment. And still one of this comparison study is in planning on nanoplastic particles. Next slide. So starting from well-defined test materials, we have developed developed or adapted measurement methods and instruments in order to be able to now go into real materials and to measure micro nanoplastic particles, the concentration, and so also the exposure we can expect for humans. And as a starting point, we also did a very comprehensive review in the beginning on the occurrence of these micro nanoplastics in humans. And as you can see, as real world samples, we have we are testing bottled water and food, but also air. We have done monitoring campaigns, indoor, outdoor, at workplaces. And also we are testing personal care products, which contain microbeads, although, you know, these restrictions from the European Commission or from the REACH regulation on these microbeads or intentionally used microplastics. And so we have also looked at this, or we are still looking. And not least, of course, we are analyzing these particles in different human tissues, in blood and excretion materials. And as part of this fate and exposure assessment, we also developed already 3 different models for fate, exposure, and toxicokinetics to show how these are translocated and interact with the metabolism in tissues and organs in the human body. Next slide. To assess human health hazards of micro nanoplastics, and AC stands for additives and contaminants, we have, as I mentioned before, we have used test materials of different size, shape, and with and without aging additives, contaminants, and leachates. And we have also adapted different, both simple and more complex 2D, 3D in vitro cell models and biomarkers in order to simulate main exposure routes, for example, respiratory or gastrointestinal tract, but also target organs, tissues, biological barriers, and the immune system. And the effects we are assessing or measuring after acute and subacute exposure include cell viability, cytotoxicity, metabolic or organal activity, barrier integrity and permeability, cellular uptake, internalization and bioaccumulation, and immune effects. Next slide. Yeah, there was, if you could go back, that was a pity because this shows the different cell lines we are using in order to simulate main exposure routes and effects in the human body. It's just to illustrate it once again. Next slide. Yeah, we also did various specific studies. One is to simulate the fate and effects of micronanoplastics in the human gastrointestinal tract. And here we found that in the GIT tract, microplastics are both transformed by the human microbiota, but also change the human microbiome. And they build a biofilm on the microplastic surface. And this is just one result and has already been published and will be continued by other studies that when we are looking more closer into and compare healthy with unhealthy patients. Healthy means patients that have a healthy microbiota in their gut. Next slide. Next slide. Yeah. So just to summarize, as also was shown by the presentation from Raymond, we also have not found any significant effects on cell viability in our assessments and on cell membrane integrity. There are, of course, exceptions where we have to look more closer into, for example, when there was a microbial biofilm or coating with contaminants, or when there was simulated digestive fluids used. These results have been different. And we also saw only minor inflammatory responses of the immune system and no oxidative stress in granulocyte-like cells, white blood cells. But this still has to be confirmed by further studies in lung and GIT cells. But as mentioned before, microplastic particles can change the gut microbiota and become transformed also, and also form a biofilm on the microplastic surfaces in the gut. And other studies, bioaccumulation and uptake studies showed that micronanoplastics can be taken up and bioaccumulated in these, both in the respiratory and in the gastrointestinal tract cells, but not in THP1 cells. These are cells of the immune system. And again, this depends on if there is a biofilm on the particle surface or an echocorona and so on. So just to also from the studies we have done so far, there is still a need to verify these in vitro results by in vivo. In our case, we use mice studies in order to come more closer to the realistic situation and look more into long-term exposure effects and using more real-world plastics. And what about leaching of additives and contaminants that are associated usually with micronanoplastics? Also look into mixture effects, how biofilms will affect the toxicity, mucus and echocorona, and also use more realistic exposure concentrations. This is also an issue for the dosimetry we are using. Next slide. So in PlasticsFatE, as also will be done in the other CAS projects, or I think most of them, also as Raymond showed, for POLYRISK, we are also trying to integrate the data we have produced and the methodology we have used into a novel human and environmental risk assessment strategy that is based on so-called IATAs, integrated assessment approaches for testing and assessments for both for micro nanoplastics and additives and contaminants. And this strategy will be also based on decision trees that are available through a web-based platform and a so-called PMCDS system. This is a prospective multi-criteria decision support system in order to guide end users during early design and production of plastics. You can see here the structure, how this will look like. On the left side and on the right side is just a little bit the schema, the scheme of this PMCDS system we will use, and this will be integrated into the risk assessment strategy. Next slide. We have also done case studies to assess the feasibility of the methods we have developed. And one case study is focusing on occupational exposure monitoring. We have done a lot of air collection campaigns, as mentioned in the beginning, at various plastic production, plastic use, recycling and packaging sites, and also one water treatment plant to look closer into how the occupational exposure of workers in these sites look like. And we have done case studies, specific case studies too, on microbial colonization of micro nanoplastics and their functioning as a vector for potential pathogens. And we already saw in the results that these micro nanoplastics, they are the surface for bacteria in marine, fresh and wastewater, and are forming a biofilm, and also that they are supporting antibacterial resistant gene transfer. So these are preliminary results that are also in agreement with other studies done in the literature. Then we also look into long-term effects by using a more simple food chain. We have looked in, we have used polystyrene and have seen that this is taken up in lettuce, in food, but that there is no further transfer to snails. So there is an uptake, but there is not an accumulation of, in this case, of polystyrene in the snails. And also we are doing, together with the other CASP projects, occupational exposure and human biomonitoring studies at workplaces in order to get an understanding of how the micro nanoplastic particles we measure in the air, how this can really cause effects in the human body. And we do this by using micro nanoplastic specific biomarkers for inflammation and oxidative stress to study these effects at realistic conditions. Next slide. Yeah, that's also the PlasticsFatE team and so forth. Thank you for your attention. I have some slides prepared for the CASP cluster, if there is time. Otherwise, then we take the next slide. So CASP is, as mentioned already by Raymond, is the European research cluster for the effects of micro nanoplastics on human health. And so this cluster consists of 5 large-scale EU-funded research projects, and the time duration is indeed similar as MAELSTROM. And but this cluster is built up by 75 organizations from 21 countries. So it's a big cluster, And the aim is really to contribute to human health relevant policies, regulation and standardization. And there, you know, these policy or strategies that the European Commission has set up. This is the strategy for plastics in a circular economy, especially, and the bioeconomy strategy. But of course, regulations under REACH, for example, this recently set up restrictions on intentionally added micro nanoplastics to products. And this will be done within CASP by providing really a robust and comprehensive scientific database and new scientific-based evidence for better preventive policies to ensure safe and sustainable handling, use, and disposal of plastic products. Next slide, please. Just you see here all the 5 projects and the main focus of the project. So Aurora is one project which is really focusing mainly on early human life phases, so placenta, fetus, pregnancy, and so on. Then we have another project, IMTOX, which looks onto health effects of micro nanoplastics, especially related to asthma and allergic diseases. We have Plastic Heal, which looks on more the health impacts and what kind of the mode of action. So what are the underlying mechanisms, and especially when it comes to genotoxicity. PlasticsFatE, as mentioned or presented before, we are really more focused on looking into fate and effects within the human body, and POLYRISK, as presented by Raymond before, also with a strong focus on immune effects. Next slide. And we have within CASP, we have built so-called working groups, which with the aim to really promote the cooperation among these 5 projects and to do this also in a coordinated manner. And here you see the different tasks that these different working groups will have. Next slide. So standardization, what is the relevance of CASP research when it comes to standardization? I think when we talk about standardization, that means we need to make our data on exposure and hazard open and fair for end users. And we know end users are a wide variety of different practitioners that really are in need of the data we are producing. Also to enable the prediction of health outcomes based on the particle characteristics or on the identification of those properties that are linked to human health effects, or for human biomonitoring studies, clinical epidemiological studies, and so on. So in CASP, we perform in the laboratory studies for method validation, harmonization of protocols, and also to support standardization, for example, within WAMAS, or also within ISO, these are just some examples of standards that we hope we can contribute within CASP. And as I mentioned before, we had this one inter-laboratory comparison study on microplastics organized within WAMAS, and this was one of the largest that has been done so far on standardized methods for characterization of microplastics. Next slide. Regulatory relevance of our research is, of course, also very important. So CASP will support regulations, different kind of regulations that are addressing environmental concerns of plastic use and disposal. And here just a short list of those regulations that you may know that are important and that need to be updated or amended, or where micro nanoplastics need to be included to ultimately reduce the emissions of these plastics and their impact on human health. Next slide. Policy relevance of CASP research. Yes, CASP, we are supposed to contribute to relevant policies, and these are grouped around the different tasks we have all within our CASP projects. That means that includes the sampling, the measuring and monitoring methods we are using in order to be able to assess fate and exposure and hazard, and finally also to characterize and evaluate the risk, and so understand also and meet the needs of our stakeholders. And the CASP findings, they will of course inform these main EU policies and legislative areas. As you can see, that includes the chemicals, plastics, food and water, and as you know, the drinking water directive or urban water or wastewater directives, they are all under revision, and partly also, as it comes for the drinking water directive, micro nanoplastics are included as one of the substances of concern. I think this was the last slide. So that was just also a very brief overview about CASP, and thank you again for listening, and questions are of course welcome. Thank you.
Thank you very much, Raymond, Rudolf, sorry, for the very interesting and the comprehensive presentation. And congratulations for the promised results that you are having in the projects. Also, especially connecting the in vitro and in vivo dedicated studies, very important. Also to fill the gaps in terms of knowledge in this area. So we take the opportunity to thank all the panelists for the very interesting and engaging presentations, especially results with novel and promising new knowledge, connecting the impacts of macro, micro, and nanoplastics from ecosystems to human health. Also highlighting our responsibility as humans to change behaviors, to change, update some of the ongoing regulations if we want a realistic wave of change. So we close now the presentation session, and we are moving now to a session with questions that will be moderated by Garry Kett. Garry Kett is our colleague in CIIMAR. He's a marine ecologist, and his previous research experience focused on the impacts of climate change and on aquaculture bivalves, marine mammal ecology, and also conservation. Garry has held with the role of Ireland's All Atlantic Ocean Young Ambassador, so engaging in high-level dialogue, ocean science diplomacy, science communication, and networking building. He joined our group, so the biodiversity group in CIIMAR and MAELSTROM project, to work together in solutions to fight marine litter with a special focus on national and international stakeholder engagement. So thank you to all. I give now the word to our colleague, Garry.
Thank you, Luis, very, very kind introduction. Yes, so some excellent presentations there in the second half of our webinar too. So thank you all very much. We will move on to the roundtable, but we'll keep it quite short because we're running over time. But I think personally, it's quite okay to run over time a little bit when the presentations are so engaging. So we might just keep it to our first two questions. Let me just share with you now. Now you can see this. So of course, no need for introductions again. You've all been introduced to our roundtable panelists as our speakers earlier this afternoon. So I would like to start by asking each of you, what do you think, which I've already put to Kayleigh Wyles, and she had an interesting answer, but what do you think are the main knowledge gaps or emerging topics in your field? So I know we've talked a lot about characterization, about weathering, and about standardization. So maybe if you'd just like to elaborate each a minute and a half each, and just kind of give us your overall objectives on knowledge gaps, emerging topics in your area of interest. So, Vanessa, maybe if we start with you, if you don't mind.
Yes, thank you. So, I think that two gaps can be, I want to speak about two gaps. One is related to sources. So working on sources, it's clear that it's not very known how many liters are transported by rivers. So we have data on beach litter and also monitoring data on floating litter at sea, but it's not clear the amount of litter transported by river. This is also because the Water Framework Directive does not foresee the descriptor for macroliter and microliter in and so the knowledge of this is not clear. And the other thing is the possibility to remove litter from the seafloor. We know that the 80% of litter ends up on the seafloor, and it's very difficult to remove them from the seafloor, and so there are big issue in developing the technologies that can potentially remove these litter from the seafloor.
Okay, great, thank you. Isabel, would you like to give your perspective on that main knowledge gaps in this discipline, in this area?
Okay, in my field, I mean, my field is environment, so of course it's looking at the impacts that this macro and microplastics have and the litter has in the ecosystems. But of course, I think the main problem and what we need probably to focus, and this is not at all my field, is how to avoid the production and really a release of these materials to the environment, to the rivers, and then to the ocean. I think this is something that is not in my field, so I'm not going to go about it very much. I mean, for me, what is would be important is to understand in the environment. And also, of course, it was mentioned in the first presentation about this human side. So we need to understand better why people have different behaviors, and, you know, how can we, you know, make them change the behaviors that led to this problem that we have. And that's it.
Perfect, thank you. And I think you also answered some of our audience questions there at the same time. People were asking, is it worth removing the litter when it can stop it being produced? And yes, we absolutely do need to stop it being produced as well. We can do both. And one of the things MAELSTROM does well is to remove while also produce policy recommendations and to affect citizen science to engage with people on a psychological level. So, okay, let's move on. Raymond, if you'd like to give your main perspective on knowledge gaps and emerging topics that you see in the research.
For sure, thank you, Garry. So the main knowledge gap that we are facing is the analysis of the particles. We need so many different techniques that are just appearing as is. They are not yet developed. As mentioned, in particular nano-sized particles, they are very, very difficult to measure because our systems physically just cannot see them. We need to do that with laser technology, which is still in development. And then we can only test the presence of particles. So how can we ever quantify the effect of particles? So that's a real challenge. With that also, the particles are so complex in composition that we face a challenge that is a classic for toxicologists, how to determine effects of combination of chemicals. Those two, chemical analysis and how to assess combination of chemicals, including particles and all the leachates, I think that's the major challenge at the current state and knowledge gap.
Thank you. Very interesting, especially from your presentation. I think looking at the weathering process and how that affects the impact that micro nanoparticles have.
Yeah, and that this was just discovered by chance or by accident. So we had the particles, they became positive, and then we looked at the surface structure, which took about half a year. And we were puzzling what was happening, and we first thought it was contaminated with microbes. That's why we checked for the microbial components, but then it appeared that the structure was changed. And that is only published by one group in that matter for oral digestion. So this could also happen if you swallow the particles. If you get them in, they may not be active when you take it from the bottle, but if they are in your gut, they may be modified to become active, which is something that is, yeah, for us, it's quite new.
Fascinating. It feels like
and it can also happen in the environment. I mean, we have all these little creatures in the sea, the plankton and the algae, but also the mussels, etc. I don't know what they do. And it would be for the future, it would be maybe not focus too much on human, because if we can do it in a simpler species, we may come to the same conclusions if we are smart enough to comprehend it all.
Okay, excellent. Rudolf, would you like to give your main brief answer on what you think the knowledge gaps, emerging topics are in this area? Oh, you're muted, sorry. Need to unmute your microphone if you can, sorry.
No, just to add what Raymond already said, we need, of course, methods that are validated and that are adapted to especially micro nanoplastics. And of course, we need to know more about what are the additives doing and what about the contaminants these micro nanoplastics take up when they are out in the marine environment, especially. And I think what is bringing together us from the human health impact side and you from those who are really taking away all this litter is these marine food web. We do not know so much. We know a lot now what we have observed, what we could observe in the laboratory under experimental controlled conditions, but we do not know so much about what happens in the real world. So the food web and human, we are at the top of the food web, but there are, of course, other organisms also on the top. So I think this would be good to have more field studies and more, you know, the biomarkers, endpoints, whatever we can use in order to see what is the real impact.
Okay, so get the feeling that there's a lot of research left to be done, a lot of challenges, but also a lot of opportunities ahead. So that's great. Thank you very much. I think we will move our roundtable short just so that we can engage with our audience and get some of these audience questions that have been rolling in. So last chance to get your questions in if you are in the audience and sitting there with your curiosities. So Luis, I believe you have some of these ready if you want to go over one or two of them.
Yes, I think we have here several very interesting questions. Starting, I think, is for Vanessa from Gregory and also John Enos. So two questions about if these removal technologies will be continue to be implemented and if they will be replicated. And also Gregory asking about why not concentrate or simply stopping plastic pollution. I think it's more a question for Vanessa.
Okay, for the first question, yes, the technology that have been developed in the MAELSTROM project are scalable and replicable. So the idea is to export this technology also in other sites to test them also for this different category of items. and in particular for the seabed cleaning platform that can pick up selectively from the seafloor or different type of items, also with a very different size, but also for the bubble barriers, obviously. And the second question, sorry, I forgot it.
You hear the sound. So why not concentrate or simply plastic stopping plastic pollution? So in the source.
Yes,
challenge. It's a challenge.
Yeah, it's a challenge. It's a challenge. We are trying to do a lot of activities to sensibilize the citizens about not to leaving plastic in the environment, but it's a challenge.
Thank you, Vanessa. And we have here a very interesting question, I think is more for Isabel Sousa Pinto, about the sources and about where the litter came. So Chifondu is asking, so what about collaborative frameworks did you put in place to address the nature of marine litter and to ensure coordinated efforts among communities?
Well, we didn't do that yet. So what we're doing is we are collecting and characterizing the litter, so we know it's coming from, and so they give this information to the authorities that are in charge of then going to the source. But since we did not have this characterization, we don't know actually what are the main sources. For instance, in the Ave River that this is a river that goes close to many manufacturing. Actually, used to be much more. Now is a little bit from fabrics and shoes and all this. So we need to see where the main source of pollutions are. So we do what some person already said, that we stop it there, instead of try to stop it at the source, instead of just trying to collect. But I think this collection, as it was presented today, it's interesting that is an activity that actually promotes the people's mental health and also well-being, because they feel well doing something. So this taken out of the marine litter has to continue, and we need to find out how to do it. But of course, our main goal, I think, should be to stop the pollution and not just get more and more technologies to take it out.
Thank you, Isabel. And we have here, I think the question is for Rudolf and also for Raymond. So as we know, we have these particles, so especially nanoparticles in our cells, in our bodies. Can we do something to potentially decontaminate our bodies?
That's a good one. We don't know. And of course, we thought about it. I mean, if you think about what you inhaled, and that's the most easy, not that easy, what we expect, that it will remain in your lungs for quite some time. It's the same as smoking particles that you get from a cigarette. That will take 10 years. And if you swallow it, most of it probably will be excreted immediately. So the uptake, what we see for oral, is not that big in a short-term experiment. So not the long-term, we don't know what that is. And if it's in, yeah, it depends a little bit. We see that the smallest particles, then the 50 nanometers that we can study, they just pass all the membranes. So they will be excreted by the kidney as well. But they will also go to your brains and whatever. They will pass every membrane. The bigger particles, I don't know, maybe they go to the fat tissue and remain there. So we actually don't know. Maybe Rudolf has an idea, but we don't have a clue. I saw a paper last week that, and that's maybe worrisome, that they found in etheric plaques. You know, they found particles. So that's worrisome, maybe.
At least in our food web study we had with snails. So the snails, they do not accumulate. They just it just goes through, and
that's good.
Will be uptaken, but not.
But if you eat a snail, Rudolf, or a mussel, you eat also the content of the gut, right? Yeah. For us, it doesn't matter.
That's true.
And if we don't take it up orally, that would maybe be a good thing. I don't know what happens if we get it in.
Yes, I think we have here.
But we can test it, and we find it in placentas, and we find it in the fetus. Oh, it is in the fetal fluid. So it's if it gets in, it goes everywhere.
Yes, indeed. So I think we have here future challenges, also opportunities for research. And Rudolf, in your opinion, is possible to contaminate our bodies?
To decontaminate?
Yes. The same question.
Raymond said, I mean, I cannot imagine that it depends, as we have experienced, it depends really on the polymer, on the type of polymer also, and also on the property, size, of course, surface, and there we still do not know a lot how this really changes the behavior of the polymer. So it may be possible that we can engineer or can make some kind of come in with some, you know, some substance that can change the behavior, the translocation or bioaccumulation of the particles in the human body. That's just idea. Yeah.
I think perhaps also important to remember from your earlier presentations that it's not just the plastic particle, but what it brings with it. So the contamination, the potential pathogen vector, the additional stressors. So a lot of research to be done.
Yes, and probably a future topic for a future webinar. Why not? So thank you all for your interesting presentations, your valuable contributions. As Garry mentioned in the beginning, these inputs and outputs will also contribute for our interactive forum. So we invite you all to register and to participate in our interactive forum, and also to a special deliverable that we have about the good practices to tackle marine litter, and also we are developing a working group dedicated to also to identify the important gaps in this topic. So I give my word back to Garry and Isabel to conclude the webinar. So thank you all.
So, this me now? Okay. No, I just wanted to thank everyone and just close. Since we are well over time, I'll just take 1 minute just to say that this was actually very interesting, and also that was complementary to the other webinars that we've been having. So here was focused really on humans and human health. So we heard, of course, that this marine litter is a purely human issue. We caused it, we suffer from it, and we are the ones that can change and can improve the situation. And of course, we heard about the impacts. Of course, these last two presentations, the impacts on our health, that is still not well understood. But also we heard something about the impacts of our mental health. And this is also important, of course. And so we don't like to see trash. And we also can, we can use this maybe to understand how to change people's behaviors. Of course, we need to change the industry behaviors. It's only not just us, but we need really to understand what makes people adhere to certain practices, and what makes people it was mentioned that the bottle, if it's nice, and people may select that one even if it's not recyclable or something like that. So how can we change this behavior? So these different behaviors that are really responsible for the situation we have at the moment. And so thank you very much. And I think we will be, so all the presentations, I mean, this recording will be available. So anyone that wants to review something or go back to this can do it. And I hope you enjoyed and follow our next initiatives. Thank you very much. Bye.